Welcome everybody to today's Math Bio seminar. We're very happy to have James Glazier here today, and he will tell us about his work on multiscale, multicellular modeling of tissue functions and how he has been using some of these tools to study viral infections in epithelial tissues. So take it away.
Thank you very much. Thank you for the opportunity to talk. I should say that Leah asked me to do a little bit of a background on what I was doing before a few weeks ago. I want to start out by saying that this is work done by, in general, a large number of people over the years contributed. For the current work, particularly Josh, Kira, Giuliano, and Sam. Sam and Kira were undergraduates in my class this semester. Josh and Giuliano are grad students in my group. Alan Cardocus is an excellent experimental biologist here at IU. TJ is a computational postdoc in my group. And then this is also in collaboration with Lutz Busch at the Technical University Dresden, who was one of the developers of Morpheus, a modeling platform, and then also James Osborne, formerly in Oxford and now in University of Melbourne. And then most recently, Richard Klemper at Georgia State University, who has been giving us some insights into the virology and drug treatment aspects of things. So I come back to these things from a very different point of view. And again, the first few slides are going to be not on virus. I became interested in computational biology fundamentally for the problem of development and self-organization. And from asking the question how organisms develop, I moved into the questions of how organisms maintain themselves and how they fail. And so my real interests have been in those kind of basic biology questions. More recently, I've become interested in whether we can use computational approaches more practically, in particular, whether there are ways of using some of these computational ideas in the context of personalized medicine. And it's certainly clear, we hear words like precision medicine, personalized medicine all the time, that medical treatment today is not very well optimized for individuals. Medicine is in some sense not scientific because you can't repeat experiments and you can't conduct control experiments on people. And of course, drugs and treatments are often rejected because they have adverse outcomes in some small subset of population. And I think long-term, the fact that medical care these days is still reactive rather than proactive is something that we're going to have to think about. And I think modeling and computation are going to be a key part of trying to advance the union of computing and medicine. And I'd say to make medicine more of an engineering discipline, perhaps. People use computing and engineering approaches all over current medical practice, personalized medicine. Of course, omics and bioinformatics are big examples of big cell atlas projects today. Lots of work, of course, in deep learning in all sorts of areas, but also things like patient-derived organoids to give you some kind of reproducibility in experiment, and tissue engineering. The kinds of things that I've worked on for many, many years have been what we call virtual tissues, which are mechanistic multi-scale simulations. And these typically are multicellular simulations. So we're talking about simulating fairly small volumes, maybe a cubic millimeter of tissue, and trying to understand quantitatively how the micro-level mechanisms lead to macro-level behavior. Ideally, you'd like to be able to then apply these insights in a biomedical context. And we've certainly worked over the years with the Environmental Protection Agency trying to understand aspects of toxicity, for example, in that way. The fundamental question, and certainly don't have to preach that to this audience, is that if you have a pathway diagram which is inhomogeneous, like A promotes B and C, and B promotes D and C inhibits D, you can't tell what happens to D when you change A unless you have some kind of quantitative model. And so the kinds of models that we build are typically 3 or more scales. We will have some kind of intracellular hairball of regulation and signaling. And one of the big problems building these kinds of models is that very often the molecular signals, which these days are very easy to measure, don't correlate very well with the behavioral level of the cells. And so that's a bit of a problem. And then we have tissue level effects of mechanics, cell movement, and long range chemical fields and signals. These days, we're also more likely to put that kind of model inside of some, again, ordinary differential equation type pharmacokinetic model of the whole animal. And I'll show one example of that a little bit later when we review some of the applications we've done over the years. Clearly, if we can get these kinds of things to work, there's a lot we could do with that. If we could really build template simulations of a tissue or an organ system and then personalize it, we could do a lot, because then we lift all of the problems with reproducibility and control experiments that make medicine so difficult. And of course, you can also try out an unlimited number of permutations of drugs, treatments, dosing schedules, and so on. And potentially, again, the thing that I would find exciting in the future is the idea that you try to design small-scale interventions to maintain health, regulate health, rather than respond to disease. These kinds of things still aren't really used that much in the clinic, although people do use them clinically. Our friends in Florida at Moffitt certainly get their methods into the clinic. People like Andy McCullough and his heart models get into the clinic. And so there certainly are places where this kind of mechanistic modeling is having clinical application. The specific kind of multicellular modeling that I do, what I'm calling virtual tissues, so far haven't really been used very much clinically, although again, the Moffitt people are certainly doing clinical trials for some of their cancer therapies that are based on multi-agent, multicellular models. If I go back in time, and again, I was asked to just do a very quick review of some of the other things I've worked on over the years, the kinds of typical questions we ask involve developmental and structural issues. We've worked a lot on blood vessel development. And so we've done some papers going back about 10 years now, and more recently looking at retinopathies. We've worked a little more recently on kidney issues, and we've worked with the EPA on chemical toxicities of the liver. Everything we do going back almost 20 years now, is built on this CompuCell virtual tissue simulation environment. And over time, I hope it's gotten a little easier to use and a little bit more powerful. One of the things that you can do now, and I'll show you perhaps if we have a little time at the end, is you can take any ODE or chemical reaction network model that you have and import it very easily into these simulations so that you can run many replicas of a given simulation inside of each cell. And that can be pretty helpful. As a background of the kinds of things that we've worked on, I mentioned choroidal neovascularization. We've worked quite a bit on autosomal dominant polycystic kidney disease, diabetic retinopathy, which was a project that we're now working on quite a bit, although I'm not going to have any new results to show you at the moment. That's now connected also to a company, which is a big a data analytics medical company like Phonomics, which is interested in AI approaches to data mining, and then the work on liver toxicity. So a classic kind of question that we would have asked until a few weeks ago would be something about choroidal neovascularization and age-related macular degeneration. This is a common cause of blindness in the elderly. And the question essentially, which we asked in this work, and this goes back to Abbas's thesis work about 10 years ago, was why do the blood vessels that supply the retina with oxygen and nutrient, the main source of supply is actually outside of the retina, although we'll come back to the ones in front of the retina in a minute. Why do those sometimes invade the retina? When they do that, things can be pretty bad, and there's been a notable lack of success in coming up with treatment strategies for this. And so we built a series of mechanistic models of essentially adhesion and adhesion failures in the retina, and we're able to simulate invasion. One of the reasons I keep showing this movie year after year is, first, it's, I think, aesthetically very attractive. Secondly, it was one of the first times we were actually able to get the time scales of the simulation right. So this process of retinal breakdown happens on the right time scales. So the micro time scales actually propagate to the macro level properly. And we did something that we had to do quite a bit with our EPA friends, which was calculate what you call a risk map, where we could say, what's the probability of particular types of retinal disorganization as a function of changes of particular biological mechanisms. In this case, changes in the epithelial tight junctions within the retinal pigmented epithelium, changes in the adhesion of the pigmented retinal epithelium to the basement membrane, here Brooks membrane, or changes of adhesion between the upper surface of the retinal pigmented epithelium and the photoreceptors. Each one of those, it winds up, can be affected by particular kinds of pathologies at the micro level. Each one of those then in turn leads to different risk factors for particular phenotypes of the disease. And so for us, that was really the first time that we ever did a simulation that gave us what we thought of as medically meaningful results. And sometime after that original publication, about 10 years ago, it was actually measured that in fact the mechanism of damage of this particular disease was an adhesion defect, which had not been examined before that paper. Another kind of typical thing that we would work on would be polycystic kidney disease. In the case of polycystic kidney disease, here's a normal kidney. This is what happens to your kidneys in this disease. It's a proliferative disease, a little bit like cancer in the sense it's a proliferative disease. However, it's not, it doesn't progress like cancer. You have inappropriate proliferation and tissue disorganization, but you don't have metastasis and a lot of the other problems of cancer. It's a moderately rare disease. Unlike most cancers, it really is genetic. If you have these mutations, you're essentially 100% likely to get it. If you don't have those mutations, your probability of getting it is less than 1 in a million. However, the issue here was that we didn't understand how these particular genes connected to the phenotype of inappropriate proliferation in the kidney. And again, there was an attempt to develop drug therapies that failed that targeted these two genes. However, there was some indications that cells that were going to participate in this abnormal phenotype expressed an abnormal cadherin, a juvenile cadherin. So again, it looked like potentially this was an adhesion defect. We built some simulations of the in vitro analog of tubules in the kidney. We're able to show that there were particular dynamics and morphologies associated with those adhesion defects that corresponded to what you saw in vitro and in vivo in the disease. And then a collaborator of mine, Rita de Almeida, developed some very beautiful bioinformatics software for analyzing this. And she was actually able to come up with some drug targets, which we tried out although at the moment, of course, all that experimental work is stopped. Another example, which is something we're working on currently, is diabetic retinopathy. So now, instead of looking at the blood vessels behind the retina, we're looking at blood vessels in front. In diabetics, you get this classic disorganization of the retina. The blood vessels become leaky and tortuous. You have loss of blood vessels leading to dropout of patches of photoreceptors. Again, this can lead to blindness. The therapies that are used either are injection of vascular growth factor inhibitors, which can damage the blood vessels that you want, as well as the ones you don't want, or something called pan-retinal photocoagulation therapy, which means that you go in with a laser and you ablate patches of the retina. So you're actually going in and destroying photoreceptors in the retina with therapy. This is a little bit like fire breaking for forest fires. You go in and you cut down trees, but instead of cutting down trees where you think it's going to block the progress of the fire, you cut them down in a grid or randomly. And of course, that means you cut down a lot more trees than you need to, and you're much less effective at blocking the spread of the disease. And so we did a series of simulations trying to come up with ways of predicting the likelihood, again, these are likelihood estimates of the dropout of particular vessels, which vessels, if they were interrupted, would be most likely to lead to progressive disease, and then to predict the optimal way to target, to protect by doing minimal damage to the tissue and have maximal probability of preventing spread of the disease. And that was work that was done by Xiaofu. Unfortunately, he graduated and went off to the crick. And so I guess he's probably locked down in London at the moment. I have a relatively new postdoc from Mexico who's picked up this work, started in January, and is now working with clinical data trying to get that project going again. Another thing that we've done, as I mentioned, with EPA over the years was looking at chemical toxicity, microdosimetry in the liver, and then trying to actually calibrate these kinds of spatial models with observed serum levels of metabolites here for acetaminophen toxicity. One of the things that came up in that kind of modeling was the idea of virtual patients and virtual populations to look at variation in susceptibility to particular kinds of toxicity. And so in the case of the kidney disease, we were modeling changes in micro-level phenotype and molecular expression that were outside of the normal. You had a cadherin that was not a normal cadherin. In this case, we're taking all of the parameters in the model, keeping them within 20%, 25%, and then asking the question, are there some, if we stochastically vary all of the parameters, are there some outliers where there are nonlinear cooperative effects that mean that some individuals, even though they're within normal range of variation, are particularly susceptible to toxicity, in this case from acetaminophen? And of course, the answer is yes. So those are the kinds of projects we've worked on in the past. And infectious diseases are conspicuously absent from that list. They're almost all structural. The liver model was a little bit more biochemical, but most of these are structural diseases. And so during the lockdown in the past couple of months, we decided, like almost everybody else, probably everybody else in this Zoom, to try to do a little bit of SARS-CoV-2 modeling. And this is work that's up on in a very preliminary form in bioRxiv. There'll be a new version of it up in a day or two. When you look at COVID-19, there's so many questions at so many levels, it's almost impossible to know where to start. Why do some people get really sick? Why do some not? What's the difference in between COVID and the flu? Why do you get this pattern in some individuals of apparent recovery followed by severe relapse? why do you have these pneumonia effects, cytokine storms, and other major adverse effects? What is it about being old that makes you so much at risk from dying from this disease? And the same question for diabetes. And diabetes is a hard one. On the one hand, I'm quite interested in diabetes from an eye point of view, but we know very little about how diabetes really works in changing human physiology. And so that's actually a pretty hard one. Coming back to this idea of virtual patients and virtual design, could we use models to triage patients better when they're sick, decide who we need to hospitalize, who we don't? Could we design better antiviral therapies, or at least optimize the one we have? And something which we're not going to get to today, and this one's the only one we're going to investigate today because we're pretty early in all of this. But another one which I'm very interested in is anti-inflammatories. Anti-inflammatories have a complicated history. I was quite interested. I even had a company back in 2007, 2008, bad time to start a company, trying to do cytokine measurements, real-time cytokine measurements. It was too early to be doing that. But the reason I was interested in that was I was interested in what we're calling here cytokine storms or sepsis. In the case of the immune system, cytokine levels fluctuate very large amounts, several orders of magnitude sometimes, over very short periods of time, over a few hours. One of the problems with anti-inflammatories is that they tend to act very strongly. If you give them too early and shut down immune response too much, you die of the primary infection. If you don't give them enough, or you give it too late, they don't work, and you then die of the cytokine storm or the septic response. And so there's a history of failed drugs. Here in Indiana, Eli Lilly is the big company. They had a drug called Xigris, which was supposed to be used to treat sepsis. It was approved and then withdrawn eventually because they couldn't titrate it right. And so to me, I think one of the biggest things that we maybe could think about doing with this kind of modeling would be to see whether anti-inflammatory therapies could be personalized appropriately. And of course, we want to know why there are differences in responses between infections by the same virus. So we're going to start with a very naive toy model of the kind we've been seeing before. We'll look at little patches of susceptible tissue. We're going to assume we're in primary infection, that is, there's no pre-existing adaptive immune response. We'll assume it's acute infection, certainly is for SARS-CoV-2. That is, you're either going to clear the virus or the virus will sweep the tissue involved. Now, if we look at the biological complexity of what's going on in this infection, it's pretty large. We have to think about the epithelial target tissue, whether it's the nose, throat, bronchi, or alveoli. There's an extracellular environment. There's also the connective tissue underneath, which we're probably going to neglect in our models to begin with. There are complex modes of viral entry, replication, spread, and removal. Lots of kinds of immune cells and immune signals. Lots of tissue damage and recovery mechanisms. And then we can't just think about the tissue. We have to think about the remote immune system and how the system responds. And in principle, we ought to be thinking about a whole body level effects. COVID-19 has issues in blood, in the heart, in the kidneys, and of course, in all of the different kinds of immune response. If we try to look at the immune response, it's complicated, and it's much more complicated than this diagram would indicate. Over very short times, you have a primary phagocytic response of sentinel macrophages and neutrophils. Actually continues a little bit longer than that. You have an antiviral interferon response, and there are other direct cell-to-cell, non-immune cell mediated cell resistance mechanisms between infected and uninfected cells. There's the early cellular innate immune response by NK cells and others. And all during this period of the first few days, the viral titer is going up. You have this initial eclipse phase of a half a day or a day, a little longer for SARS-CoV-2. Then over starting about 5 to 7 days, you have major adaptive immune response where cells are being triggered by dendritic cells going to the lymphatic system, triggering production of T cells and other cells in the lymph nodes, and then coming back. And that's really when you start clearing the virus, assuming that you're lucky. Tissue damage starts about 2 or 3 days and increases as the T cells come in. Eventually, your B cells start producing antibody, although by the time that's happening pretty well, the infection is over. Now, if you were going to have complications, there'd be a whole other set of graphs of this kind. And this will vary by the tissue, the virus, and the individual. This is general pattern of immune cell response. If we want to think about immune response component, we should at least think about the phagocytic component early, which controls viral loads early, interferon response and viral resistance also early, systemic cytokine signaling, which is relayed by immune cells, amplified by immune cells, recruitment of NK cells and others, signaling to the lymph nodes by cells, a return of adaptive immune cells, and then B cells and antibodies, and then the complications. The blue ones are things that we're going to include in our base model. The black ones are things we know are important but haven't put in yet. Okay. If we look at viral replication, there are a lot of data in vivo, in animals and humans for viral replication in influenza, for example. Not quite as much for SARS-CoV-2. What you see is an interesting pattern, which is that the viral load in the organism, you have this very rapid exponential increase of virus. First, if I go to the bottom slide, so this is in culture, you see this eclipse phase for SARS-CoV-2 of about 6 hours, followed by a 1,000-fold increase in viral load over the next 6 hours, and then saturation within 2 days. And for influenza, that's really when you get sick, when the viral load peaks. And then the viral load goes down gradually, even though the number of infected cells is going way up, the total amount of virus goes down. And then when the adaptive immune system comes in, about day 7, the viral load collapses and you clear the virus. And so even in vitro, where there are no immune cells, you still see a saturation of production of virus after here about 12 hours. And that's a little bit surprising because it means, and the number of infected cells in this experiment was increasing, but the total amount of virus being produced per cell was going way down. And so that means there's also saturation of virus production within the cell. Cell death doesn't seem to kick in until a little bit later, so it's a little bit mysterious what's causing that. We can discuss that in more later. It's not in our models in any great detail. Tissue damage starts around 1 or 2 days, cytotoxicity due to the virus. There can also be antiviral signaling-induced apoptosis. Certainly things like natural killer cells and CD8 plus T cells will kill infected cells. You can have some bystander effects in some cases. I don't think those are probably very important for SARS-CoV-2. You can have immune cells that secrete oxidative species and kill not only the direct cells they're synapsing to, but their neighbors. And then if you wanted to have a more complete model, you'd need some kind of tissue recovery and cell proliferation. So the minimal thing we've got to do if we're going to model all of this is include some representation of objects, virus, epithelial cells, immune cells, and some kind of representation of what's happening in the systemic immune system. Viral replication itself is very complicated. There's this nice commercial picture of SARS-CoV-2 replication with a nice list of inhibitors that target various aspects of that replicative cycle. We're going to simplify things a lot. The only thing that really matters in viral replication from the point of view of a mathematical model is the replication of the viral genome, because that's the one exponential term in the replication model. Everything else can be amplifying, but it's a linear amplification. It's a linear feed forward. Viral replication actually exponentially increases the capacity to produce virus. There also has to be some kind of saturation. Here we have an arbitrary Michaelis saturation term because the capacity of the cell to synthesize virus is limited, whether that's by ATP availability, protein availability, endoplasmic reticulum capacity, number of ribosomes, and many possibilities, but there's always some kind of saturation, and that's respecting the curves that we saw. You can get an estimate of the eclipse time by looking the reciprocal of all the rate constants and adding it together. You can get the time scale for 10-fold increase of the virus there. You can estimate the maximum rate of production of the virus there. So just finding the fixed points tells you basically how this model behaves. And you can compare the time series of a model of this kind. The ratio here in orange, it's viral RNA, and red is protein. That ratio is essentially arbitrary. It's controlled by one of these parameters, these feed-forward linear parameters. But what we're trying to do is replicate the fact that over a period of 6 hours, the viral amount of virus stays low, comes up quite rapidly over the next 6 hours, and then it gradually saturates. And this is again happening in vitro, so this is respecting what individual cells are doing without immune system involvement, although with interferon signaling, which we haven't included. As I mentioned, the actual physical structure of the epithelium is very complex. We have a very simplified model where we have a tissue that consists of epithelial cells, immune cells of the extracellular environment. To begin with just 3 chemical fields or spatial fields, a density of virus, an oxidative agent secreted by activated immune cells, a single cytokine signal. And probably the biggest weakness of this model is that we've simplified all of the signaling of cytokines and chemokines into a single number. And then this very simple model of immune cell recruitment, which we're calling a lymph node. Within that, we've included some models of viral replication, cell death caused by viral replication, contact toxicity from activated immune cells, oxidative agents, viral release and the spread in the environment. The immune cells have this contact toxicity. Activated immune cells chemotax to the cytokine. They can be activated by the cytokine. They can secrete the oxidative agent, and they're brought in and taken out of the tissue recruitment by the lymph node systemic immune system model. There are 4 basic cell types for epithelial cell. 3 of those correspond to something biological. One of them is an artifact of the way we represent. You can have an uninfected cell, an infected cell that's not yet releasing virus, and a cell that's releasing virus, and it can die. And so we have a variety of behaviors that these cells can have. Viral internalization. We don't completely prohibit superinfection, but we do have a concept of receptors. And when the virus internalizes when the cell internalizes a virus, it reduces the number of surface receptors, so it makes it harder for the cell to internalize additional viral particles. Viral replication, viral release. Infected cells produce cytokines. They can die from producing too much virus, and all kinds of cells can be killed either from the diffusible oxidative agent secreted by activated immune cells or by contact with immune cells as well. We have 2 kinds of immune cells. This is some kind of very coarse aggregation of a lot of different immune cell processes. We don't have phagocytosis here, which is probably the single biggest thing besides interferon that we're leaving out. The inactive immune cells can move randomly, they can contact kill, and they can activate. The activated ones chemotax in response to the cytokine, which is a chemokine as well for us. They can release the cytokine themselves, a relay. They secrete this toxic oxidative chemical, and they also have a time over which they stay activated. They inactivate after some amount of time, which may not be that realistic in this particular model. The immune system is aggregating all of that cytokine information into a single number, which is an S, which is the pro or anti-inflammatory state. If S is bigger than 0, we add immune cells to the tissue. If S is less than 0, we remove immune cells from the tissue. And it's a little bit of a complicated set of ODEs and some events that determine S. There's a delay so that the recruitment takes some amount of time. So the number of immune cells we supply to the tissue reflects the cytokine level sometime before, with the idea that that's reflecting a bit of the adaptive immune system delay. On the other hand, we know things like macrophages and neutrophils are recruited pretty fast by cytokines, and so averaging those is maybe not such a great idea. There are a lot of parameters. We didn't really make a strong effort to parameterize this model because it was a proof of concept. We do know that there seems to be a strong dose dependence to the time course of the disease. And that means that the immune response and the viral spread have to more or less balance. And so we respected the very basic issues of timescale of viral replication and spread of infection and clearance, but we weren't too concerned about the rest. We wanted to have immune system and viral spread more or less in balance, but since we're not modeling the later phases of the adaptive immune system, we did not want our base model to have the immune system clear the virus. And so we picked a set of parameters that did that. If we look at the viral replication by itself without the immune cells, we start out with a single infected cell in the middle. It, after a while, releases virus in the second row. That virus then goes on to infect neighboring cells. Red cells are secreting virus, green cells are infected but not yet secreting, black are dead. And in our little tissue patch, we get a nice propagating ring of Fisher type that sweeps the tissue and kills all of the cells. We can look in time at the number of infected cells, the number of virus-releasing cells, the number of dead cells. The orange curve is the number of uninfected cells. And also then the viral load versus time, and the cytokine level versus time. And we see that this peaks, these are actually lined up. The viral load peaks around 10,000, 12,000 minutes, a little bit late, probably for influenza, probably not too far off for SARS-CoV-2. There's not the kind of, it's a short latency period, maybe not enough. Now we're going to turn on the immune system. And here in the second row, we see the immune cells coming in. Those immune cells kill a lot of the infected cells. They also relay the cytokines, so cytokine levels go up a lot. But we still have a situation where all of our cells become infected. Eventually, all of the cells die. And what we see here at the end of this simulation is that the viral load is going down, not because the immune system has cleared the virus, but because all the cells have died, so there's nobody producing virus anymore. One thing we do see, though, is that the total amount of virus is kept down from the case with no immune response, and the peak happens a little bit later. So the immune system is slowing the infection, even if it's not able to contain it. We could do parameter sweeps. There are a lot of parameters to sweep. In this case, we're sweeping essentially the strength of the immune response, so stronger immune response to the right, and the faster viral internalization. The bigger the viral internalization constant, the more easily cells are infected for a given amount of virus. 10 replicas per parameter set. And we see two basic reasons here. In green, we have situations where the immune system is able to contain the virus. So in these situations, all of the infected cells are eliminated, and all of the virus essentially is eliminated. Here on the right, we see the virus goes to 0 at 2,000. From the bottom right, we see something interesting, which is the virus goes away, and then a few cells get infected by residual virus and produce a virus, and then the virus goes away again. We call that recurrent infection. Down at the bottom left, we see the same kind of sweep we saw without the immune control. All the cells get infected, the viral level goes up very rapidly with a little bit of saturation around 5,000. And then it's starting at 10,000, the cells are starting to die off. The virus is decreasing because the cells that are producing it are dying. If we go to the top, we see more interesting behaviors. Sometimes we see sweeps of the system, as we would see for fast viral internalization. At other times, we see something where the virus is eliminated. And so we have coexistence of very different outcomes for different replicas. And so that means that going to a stochastic spatial model is really important. And so this stochasticity of outcomes for different parameters, I think, is critical. If we zoom in and look at our region where the virus sweeps, we see less variation. Top right corner, we see some. Basically, we see the same pattern in each case. This white one, one replica did something different. If we look at the immune state, we see if the immune system is very weak, nothing happens. As the immune system becomes stronger in its response, we get a big peak of immune response. A lot of immune cells come in, but it comes in too late to prevent the sweep of the tissue by the way. If we look in the other side, in the top right, where the virus is under control, the number of infected cells is always few. We get a few infected cells, they go away. In these white areas, most of the time they go away, but sometimes they come back. And we see this kind of recurrence in this white area, viral clearance in the left, all the way to the right. In this case, all the way on the bottom here, second to left most column, bottom, we see one case where the immune state seems to be taking off at the end, corresponding to an increase of virus at the very end of the simulation. In these intermediate cases, things are a little bit more interesting. If we look at viral load, we see some replicas where the virus comes in and disappears, other ones which we get this sawtooth of clearance followed by recurrence, some where we get gradual increase in viral load, some in which we see saturation of viral load. So it's a very complicated zoology of possible coexisting states for some of these parameter sets. We're just beginning to explore that. We tried to classify the responses. These aren't real hard phase transitions. They're more of a continuous gradation from the sweep with no immune response through widespread infection, which is our reference, slowing of the infection, more slowing, to containment of the infection where the virus doesn't go away, but there are only a few infected cells at any time, that's bottom left. Recurrence, where we get rid of all of the infected cells, but one or two cells get infected again from residual virus, and you get a periodic pattern of that. A case where the immune system successfully clears the virus entirely. And in the case where the infectivity of the virus is very low, you can have an initial infection where everything stops, where there's essentially no, the infection doesn't take. There are other kinds of parameters you could probably find, but that's the basis. My real interest in this was to try to see whether we could do anything meaningful about therapy. Clearly, with a toy model like this, we can't be serious about therapy, but it happens that the RNA synthesis step is a parameter in the model, because remember that the most important thing about viral synthesis is the replication of the viral genome, because that's the one exponential amplification. Remdesivir and other RNA polymerase inhibitors block that step. And so we can look at that. That means that this drug action actually corresponds to a parameter, a single parameter in the model, which is very nice. We know that antivirals have a problem. That is that as you wait after time of infection, their effectiveness goes down rapidly. And there's always a trade-off between how much of the drug you use, its potency, that is a product of dose and drug efficiency, versus effectiveness, what it does clinically. We want to use the lowest possible dose to minimize side effects. No drug is completely without side effects. In our very simple model, we'll assume that the drug is given and kept into the system. We're not going to model washout and dosing schedules, but that's our next step. And so we can do the same kind of parameter sweep that we did before. We find that if we give the drug early, so earlier is to the left, later is to the right, and by early and late, we're referencing the time of maximal viral load in the untreated simulation. So early means we give it while the amount of virus is still increasing rapidly. Late means we give it about the time the virus would peak on its own. Red means that most of the outcomes are that the virus sweeps all of the cells. Green means that most of the cells are uninfected at the end of the simulation. White or light shades are intermediate cases. We'll notice that it's most important to treat early. Even at quite low potencies down here in the second to bottom row, if I go all the way to the left, it's still green. That means if I reduce viral genome replication by only 20%, if I treat early enough, I can still prevent the infection. That to us was rather interesting. For some of these, especially at later times, there's a lot of stochasticity of outcome. And so I'll zoom in on one of those cases. This is a perfectly effective treatment given relatively late. And what I see, if I look at the total number of uninfected cells at the end of different simulations, sometimes I have no uninfected cells at all, almost no uninfected cells. That's this bottom purple line. Sometimes almost all of the cells are uninfected. That's the top green. So there's a huge variation in outcomes, negative, positive, and intermediate for the same parameter set, even with just 10 replicas. And that's happening essentially because there's a lot of stochasticity in early infection as to how rapidly the virus spreads early. And that means that there can be some uncertainty about the time at which viral replication peaks. And if you're lucky and that's delayed even a little bit, there's a critical window of time where coming in near that peak, if you come in just before that peak, the treatment is effective. If you come in just after that peak, the treatment is ineffective. And so you can have a situation where most of the time, if you imagine a patient, most of the time if you gave them this drug at that time, it wouldn't work. But in a few cases, it might still work. Now, of course, in reality, if the virus is moving from tissue to tissue, treating late might still be very beneficial. And certainly there are clinical studies which suggest that it is. Not very, but somewhat beneficial even if given later. But this gives you some way of exploring why you get that stochasticity in outcomes in the patient. So the main results are first, early treatment is better, which you didn't need us to tell you. Secondly, that even very low dose treatment earlier is better than higher dose treatment even a little bit later. That maybe is a little more surprising. And lastly, that even for a fairly late time treatment, if you treat at high levels, a few replicas benefit. That probably is also clinically unsurprising, but it's a nice result to get from the simulation. There are a lot of things that are missing from the simulation, almost everything really. The spatial structure of the tissue was very regular. All the cells could be infected, which isn't true in the lung. So we've done simulations now with more realistic geometries and distributions of infectable cells. We're beginning to do simulations where we include interferon-induced viral resistance. This is Sain's model of that, classic OD model of that. We're including phagocytosis, and we're beginning to include additional cell types. Of course, we could do better with viral internalization, although I don't think viral internalization is probably a rate-limiting step for most of these situations. Of course, there are all sorts of intracellular signaling. There's complicated transport. We're assuming diffusion of virus, but in the mucus, there's complicated transport. We don't include any kind of tissue recovery or humoral adaptive immune response. You'd like to put this inside of the whole body. If you're going to do drugs, you want to be able to understand how the drug's delivered and cleared. And of course, you'd like to be able to calibrate things to real viruses and tissues. In the case of real calibration, these kinds of spatial models traditionally have had some issues with that. We've been developing a very systematic workflow, which is very, very easy to implement in CompuCell, where we can take an ODE model that's been validated against experimental data. In this case, we're working with Amber Smith, who has some nice models of influenza infection and clearance, 5 ODE model in this case. We can implement that 5 ODE model in the CompuCell spatial context, exactly replicate it modulo stochasticity for low rates of infection, because just like with Gillespie, when you have very low concentrations of agents, very low numbers of agents, then the stochastic models will diverge from the ODE. As soon as you have a large number, you can exactly replicate. Once we've done that, in a mixed model, it's now an agent-based, but we haven't really turned on the spatial component. We can now turn down the diffusion constants, and now any difference in result we see is guaranteed to be due to the spatial component, because we've shown that if we take the homogenization limit of turning diffusion constant up, we exactly recover the ODE models. Since it's easy to validate the ODE models parameters against experiment, because there aren't so many, we can then take the validated ODE parameters, use them directly in a CompuCell model, turn them directly then into spatialized models, and be sure that everything we're seeing is in register with the experiment. And so that's where we are now. That's something we don't have in our paper yet. That'll be the next thing we put in. So that's as far as we've gotten. Clearly, this is a big job. We'd love help. Everything we do is very preliminary. And so if anybody's interested in talking to us and working with us, we'd be happy to do it. We certainly are not, except with the exception of Richard Plemper and now Amber Smith, we're not people with strong virology and immunology expertise, and so we'd love to learn more about it. In particular, also, if anybody has ODE models, we do have this very systematic and simple way of translating ODE models into spatial models, and we'd love to teach people how to do that and work with them on it. And so those are the main things I wanted to say. We're going to be trying to teach our CompuCell course this summer virtually. You can find out about it at our website, CompuCell3D.org. There's some new releases of CompuCell that let you run it on Amazon Cloud and those kind of things. Background support. Have to disclose that I have 2 basically empty companies that I set up to try to do some commercialization of the diabetic retinopathy and the kidney work. And also, back in the fall, I took a flyer by putting some of my pension plan into a few shares of Gilead stock. And since I have the word remdesivir in my talk, I have to disclose that I own a few shares of remdesivir of Gilead stock through my pension plan. Thank you very much for being so patient. I don't know, applause doesn't work very well at a virtual meeting, does it?
No, it really doesn't. But we can still try. Questions?
Yes, may I
Hey,
ask
John,
a question?
Cindy,
goodness.